Thanks, Bob. I only got the brief of this sort of yesterday, but yeah, in terms of the Australian perspective, of course, I'm not going to know all the facilities we've got in Australia, so it's a little bit difficult. So I've tried to just sort of, I guess, look at our situation, what we have, to try and spark a bit of conversation around what we might need and what other facilities. So we've heard some talks today. Darren's talked about the facilities in terms of universities and their potential to in terms of being able to add to botanical gardens and that sort of thing. And, you know, there's a range of different groups doing research in different spaces. We've got different facilities and different expertise as well. I just wanted to go through some of the facilities I guess we've got. We're fairly blessed in that we've got fairly new facilities in some of our sites, but we've also got a lot of older facilities as well. And some of those include, you know, massive areas of properties that have been used for various trials and that sort of thing. So when we started doing a lot of this work, we've done a range of different species where we've done screening work, and we've been blessed by having controlled environment rooms, massive rooms, brand new, because you know what the old ones do, and those old, rickety old things, so they never can maintain the temperature, you can't get the right humidity, but these have been perfect. So we've been able to screen a lot of material and look at resistance, both with rust and other fungal disease we've been working on. At that same site, we've got glass houses, we've got shade houses, we've got biosecurity systems all set up there, even shower in, shower out systems. So we're quite blessed in that way. We've also got other sites where Tracy is up in Gympie. We actually grow a lot of our material there, establish it, bring it down to Brisbane, do all what we need. We've also got another site out at Redlands, and it's shared with University of Queensland there too. Massive glasshouse facilities as well as fields facilities there as well. We do have seed storage facilities, mainly for things like eucalypts, because our drive was in plantation eucalypts, and it's not really obviously set up for a lot of these fleshy fruited side of things, but it's still a facility that's worth maintaining. The problem is, you know, these aren't all free. Even though we're government, we have to pay for every single one of these facilities when we use them. So we have to get external funding in to be able to do projects that utilise any of the pieces of equipment. Then I started to look and think about, well, what expertise? And I know we've touched on, there's been pathology, there's been breeding, there's been botanical expertise in here, and we're blessed, I guess, with a good group just within the forest health perspective. We've got a whole raft of other people that work across agriculture and horticulture. But in our field, in our area, just in DAF and Brisbane, we've got diagnostic capacity, we've got people with field experience like myself, and then we've got those with molecular experience, and we've heard the talks that have gone on about the work Louise that has been contributing to us in that space. We do have tree breeding expertise through our collaboration with the University of Sunshine Coast, but that's moving away from forestry. David Lee is now working, I think, in ginger, so there's less and less work in that tree space. But there's need to reinvest in that space. There's nobody coming up behind him in that area. We've got a lot of technical expertise. We're not replacing that technical expertise as probably as quickly as what we should be, or supporting that expertise. And I wanted to put up entomology up there, and you might be thinking, well, why do we need entomology? We've talked all about the decline of the species and all that sort of thing. We haven't talked about all those insect species, the pollinators, even individual insects that might rely on those individual hosts. We know on Rhodamnia maydeniana that it's disappearing. We had a really quick look at sort of some of the tiny little mites and things that might be present, and there was one totally undescribed species on that Rhodamnia maydeniana which is disappearing. So what sort of other things that are actually affected? What are the flow-on effects? So we do have a team, but we just haven't sort of interacted in terms of any formal projects, I suppose. I've actually done more work with the New Zealand guys in relation to bees collecting spores and the impact that might have on hive viability and that sort of thing. So we do have multiple field sites, as I said before. When we were doing all the hardwood eucalypt trials years ago, there's trials in there's sites in North Queensland, there's sites in Central Queensland, and then closer down to the southeast Queensland. Some of the more active ones have been in Traviston, which is just near, I guess, that Sunshine Coast, the western area of Sunshine Coast, and we've recently used that, and I'll run through some results in a second. The Redlands site, as I said, might be perfect for us because it's on that coastal area. and North Queensland. Walkerman's probably a bit iffy because it does have a lot lower rainfall than just down the road in the Mareeba or in the Cairns area. But there are those sites available. Again, it's just a matter of investing. We need the money to actually use those sites. They're not free. We need somebody obviously preparing those sites, maintaining those sites. There's all the herbicide programs that are required if we're going to set these programs up. So I just wanted to talk to you, I guess, briefly touching on the screening. This is sort of doubling up a little bit. As I said, a lot of our initial focus was on eucalyptus and Corimbia. We've done glasshouse only. We haven't pushed these out in the field trials. That was just trying to get an idea of what the susceptibility levels were, what was going to be the risk in terms of plantation industry and even the native forest logging processes and that sort of thing. And we looked at that because we have well-structured populations of those. We're able to look at that at a provenance level. So we're looking at a north-south distribution, then an east-west distribution, and then also at a family level. We found resistance pretty much in all families, but there were some provenances that were more susceptible than others. And in the case of, say, Eucalyptus cloesiana, there was a nice pattern where the inland provenances were much less susceptible than the coastal provenances, which is quite odd because you'd think all those coastal provenances have co-evolved with a lot of different pathogens over time in comparison to those western ones. One thing of note, and one thing when we're looking at all these species, is that when you're selecting or breeding for rust resistance, we have to be conscious of all the other fungal agents or even other insect pests that might be affecting those species, because we did find in Corimbia and in Eucalyptus globulus, rust resistance often meant that they were susceptible to the endemic pathogens. So we did have individuals who were resistant to both, but we have to be extremely careful that we know what's going on there. Same, yeah, globulus with Tradosferia. So the populations that were selected for Microstrella leaf blight disease, they're actually more susceptible to rust than those that were considered resistant. So this is work done by Emily Lancaster on the Backhausia. She's just had her PhD accepted, so we can start sending out the information soon. But the main thing I want to show here is the sites where we set up. We were lucky enough that one of the growers allowed us to set up a lot of the trials. This germplasm it represents the whole collection of Backhausia citriodora across its distribution, and includes a whole range of different provenances. And there is a bit of variability in terms of phenotypic characteristics. And we did comparisons in terms of what the with the main commercial variety. So in here, there's one thing I wanted to point out, but maybe just on the second one. So these sites are still going. What I want to point out, I guess, the value of having two sites is that it was a site by provenance interaction. So the Traverson site's slightly drier than the Dunoon site, and so the disease levels at the Dunoon site were much higher, so there's a much higher disease pressure in there. I mean, it's great information in terms of telling us, I guess, from an industry perspective, that the higher rainfall areas are obviously higher risk in terms of getting good production in comparison to the Traverson site. The other major thing I guess I want to talk about, and Tilly can comment and AJ can comment a little bit more on this. So, you know, we've talked about taking this material into various places, moving into different botanic gardens and that sort of thing. But there are places where we can't take material off country. For example, perfect example, I mean, Gari's a nice island place, so it's a little bit easier to define the boundaries and that sort of thing. But under bachelor law, we can't actually take that material off the country. So we have to look at how we might establish a program on country, whether that's be through seed collection or whether that's, you know, whatever way we think we might be able to manage the issues. But maybe we should be also thinking about this in a different way. So rather than actually collecting and storing material and screening it, we should be looking at ways of how we can make country more resilient, I guess, to the disease on the site and nurture that natural regeneration in these sites. But we need to think about that, because it's not this case that we can just take all this material, put it somewhere else without consideration of law of the Indigenous people. So this is just to demonstrate some of the work that we did when Tilly was working on Gari. So this is Melaleuca quinquenervia, where we've actually identified individual trees that we know are showing disease tolerance or resistance. based on plots we've set up and return surveys. So yes, we know there's a lot of susceptibility within those populations. We know there's individuals. So we've identified those individuals to go back and get seed from those. Unfortunately, because it was a project, funding ended, so we haven't gone to that next stage of actually collecting any material. But anyway, that's all I had to. So I think, Karen, you might be next up.
All right, Geoff seems to be much better at me at putting together PowerPoints on the fly. So mine is quite generic, but just wanted to look at what facilities and resources we have ready to go for resistance breeding that we could use. And I think I debated where to put this slide, but I think one of the most important resources that we have is our people. And the information that they've acquired over the last decade on how to propagate, how to maintain, how to spray, how to grow these species. Geoff's experience in field trials, and then we have Craig's experience in finding plants in the field and collecting the material to send to us. All of that is very useful information that we can draw on to start our program and to continue it. And it's not working. Oh, there it is. It's not working here, but it's working up there. Let me just go back a bit. Okay. Okay. The next big asset that we have is the plants. So we have really good collections now for Rhodamnia rubescens, for Rhodomyrtus sidioides, for Lenwebia species main range. And those collections are well maintained, and so we can use them for breeding right from now. The thing that goes along with that is all the and the other thing we have is all the genetic data that we've accumulated. So we have information on the distribution of the species, information on how the genetic diversity is distributed across that distribution, how much of that diversity is held in our collections, and which of those individuals in our collections are showing resistance, particularly for Rhodamnia rubescens, not so much for Rhodomyrtus yet, or Lenwebia, so those ones still need to go through a screening process. But we have really good information now for Rhodamnia rubescens that we can use to start a breeding program. Nurseries, we have good nursery facilities at the Australian Plant Bank, but there are also nursery facilities in many of the botanic gardens that we can liaise with. So there's the Australian National Botanic Garden, Royal Botanic Gardens Victoria, Botanic Gardens in Queensland. All of those have the facilities and the experience to do some work along these lines. So we're set up with the space that we need at the moment for the collection size that we have at the moment. We can maintain and use for breeding purposes at the Australian Plant Bank. There is always the issue, of course, of keeping those plants in good health, which can take quite a lot of work and quite a lot of spraying, as Veronica can attest. But we have the material there, and we know how to treat it, and we know how to keep it healthy. The next thing we have is seed banks. We have seed banks in every state and territory. We can use the seed that's already held in those collections as a starting point for material for breeding programs to screen. We can also use the seed banks to hold seed that we get from the breeding program so that we can use it and put some seed out, see how it goes. If that doesn't work, if it fails for some reason, then we've got more in the bank held ready that we can repeat the experiment. So seed banks are always really useful for that. And of course, we have the expertise within those seed banks for collecting the seed, cleaning it, processing it, and storing it in the right way. Tissue culture. Why has that not come up? So, tissue culture is not necessarily necessary for the breeding program, but it is something that we could potentially use if we get a number of individuals and we want to do field trials at different areas with those same resistant individuals. We could potentially multiply those up with tissue culture. So it's an option, not necessarily a necessity, but it's good to have that there as an option. We have some field trial sites at Australian Botanic Garden. Might need to twist John's arm to use more of the land for those sorts of things, but that's one potential site that we have for doing some planting out for the field trials. We also have other regional botanic gardens that we've been working with who might be interested in contributing to that program. And I don't know, Geoff's got some fantastic sites that maybe we could latch onto as well. And monitoring tools. So this is the tool that Craig was talking about in his talk, the EpiCollect 5, which is already set up for recording information on disease incidence for the myrtle rust and what sort of symptoms you're getting and how it's affecting your species. So that's something that could be used if we're distributing these over different field sites to look at performance under different conditions. Then we can teach people how to use this, and it's quite an easy app to download and to use. And the final thing, of course, is networks and research partners. So our partners with the CHARBAG, the Council of Heads of Australian Botanic Gardens Australia, New Zealand, the Australian Seed Bank Partnership, the Australian Network for Plant Conservation, lots of people that we're already working with, and universities, of course, lots of people that we're already working with that could be called on or to help with finding sites for doing field trials and for helping to monitor those plants. So we have lots of resources ready to go. I'm quite excited. I think we could get started tomorrow, except that you've all got a field trip to go on, so maybe next week.